I would like to welcome you to today's webinar titled Planning and Implementing Large Cardiovascular Outcome Studies, Opportunities and Challenges. This solution provider webinar is brought to you by DIA in cooperation with CVOT. CVOT is the preeminent provider of clinical development services and specialized trial design and analysis software for pharmaceutical, biotechnology, and medical device organizations of all sizes. After the webinar is complete today, you will be automatically taken to a page to complete our brief online survey. Please take the time to evaluate this webinar. DIA is committed to developing quality educational programs that meet the needs of its members, and your feedback is greatly appreciated. The results of our online surveys are used to plan future programs. Questions and answers will be taken after today's presentations, but please feel free to submit your questions at any time by using the question and answer box. The question and answer box is located in the lower right-hand corner of the WebEx screen. The faculty for today's webinar is Zoran Antidiabetic, Senior Director, Strategic Consulting, CVOT, and Dr. Paul Strumpf. Dr. Strumpf is Vice President, Clinical Development with Lexicon Pharmaceuticals. At this time, I will turn the webinar over to Mr. Antidiabetic. Enjoy today's webinar.
Thank you, Ben. And again, the title of our today's webinar is Planning and Implementing Large Cardiovascular Trials. So I'm just giving you a few seconds to read this disclaimer. And then this is the outline of today's webinar. First, we will provide you with some background. Then Paul Strumpf will present you with medical considerations as a background to this presentation, and then I will go a little deeper into strategy and design necessary to accomplish objectives of these trials, and then finally, the final step, I will be getting into sample size and duration and some implementation aspects of these trials. So as I said, first a few words on the background. The biggest event that necessitated these large CV trials was the FDA guidance published in 2008 for diabetes, evaluating cardiovascular risk in new antidiabetic therapies to treat type 2 diabetes, resulting from some issues experienced with some drugs at that time, and Avandia in particular. Two key requirements that are somewhat technical and written in somewhat of a statistical language is that the upper bound of two-sided 95% confidence interval of the risk ratio of treatment over control has to be less than 1.8 in order for drug to satisfy the requirements to get approved by the FDA. And in order to accomplish this, the sponsor would be required to combine the data from all phase 2 and 3 trials, cardiovascular data from these efficacy phase 2 and 3 trials, and if that information is not sufficient, the sponsor would be encouraged to initiate a separate outcomes trial, cardiovascular outcomes trial, in order to accomplish this requirement. And then for the post-marketing, there will be an additional post-marketing requirement, and that is that the upper limit of the confidence interval of the risk ratio of treatment over control is less than 1.3. So I know that we have a very diverse audience, and while my background is in statistics, I will definitely not be speaking in very technical language, and over next hour and a half, over the course of next hour and a half, I will do a lot to illustrate what exactly these requirements mean and how to define strategies to accomplish them. As a follow-up to this guidance, the sponsors developing diabetes products, and there was actually a good number of products that had to comply with this guidance. Paul will get into more details there. Sponsors are now conducting these large clinical cardiovascular outcome trials because efficacy trials are definitely not sufficient to accomplish these requirements. And not only are these requirements made now for diabetes, but actually the FDA is now making similar requests for a number of other indications as well. They are not exactly the same as the guidance for diabetes, but similar. And I think this presentation will address many of these issues that are specific not only to diabetes but beyond. Just so that you can follow this presentation better, I thought it would be good to give you a little bit of an outline, or describe the presentation flow. So we will begin with medical considerations to put this whole presentation into a broader context. Paul will be responsible for that part. Paul will present that. And then, since cardiovascular trials are event-based, the whole strategy and design, everything that I will be presenting, that part will be discussed in terms of number of events. And again, this is where things can potentially get technical, but I will certainly do my best to present this in a way that's understandable to everybody attending this call, this webinar. So once we decide, define the strategy and design based in the terms of number of events, then we need to define what does that mean in terms of number of patients, in terms of the length of development, in terms of timing of approval and things of that kind, and that will be presented in the last part of this webinar. Now, as usual, in order to demonstrate these strategies, we are using some let's call them case studies, and for these case studies, we had to develop some assumptions, and I would like to ask you not to focus on assumptions themselves. That's not the primary objective of this call. It would be much better to focus more on concepts and really, the strategy of how to accomplish what is required by the FDA nowadays. So that being said, this is really an outline of presentation and what you can expect to hear over the next hour and a half or so. And now we will switch now to Paul Strunk. And Paul and I had pleasure to actually work on development of a couple of these programs jointly with Paul. So we collaborated on this, and I'm sure have very good common understanding of or can present this even jointly in some way. So again, I will switch to Paul, and Paul will give you details about we'll present the medical background here.
Thank you very much, Zoran. And I think we're now going to go to slide 8. And while we're actually going there, what I wanted to talk to you about is the fact that, as in most clinical development, there is no one correct way to design a trial, and the cardiovascular outcomes trials are a very good example of that. I'll be showing a slide later that demonstrates the variability in size and also endpoints. Within a study that's chosen for a certain design, there are many considerations, such as what MACE rate should be assumed, what population should be studied with what MACE rate, and many sponsors have used different strategies to get to a MACE rate that is around 4% or so. Additionally, there is the question of total number of events, which Zoran will go over in great detail, that are required to meet that 1.8 threshold for the FDA to accept the new drug application. And some sponsors have tried to meet that with small studies that are of rather long duration. Others have met that with very large studies that are of short duration. And the actual strategy depends on trade-offs, like most of clinical development. So what Zoran will be presenting to you today is a spectrum of different choices that can be made and have been made by sponsors, and we just want to stress that what is right for one sponsor obviously is not right for all. As we go to more concrete discussion, I wanted to talk to you about the current situation in cardiovascular outcomes trials, describe the cardiovascular outcome landscape, if you will, some strategies for acceleration of NDA filing, and actually the possible unintended consequences of going down various roads. And the idea of serving two masters, that is to have both the number of events that are required for the NDA to be accepted, and now the consideration that the cardiovascular outcomes trial, if it is a separate trial, is the largest trial in a program and probably has the largest impact on total safety exposure for requirements. As we advance to the next slide, talking about the current situation, this news is not new. There is a worldwide epidemic of type 2 diabetes. According to the International Diabetes Federation, in 2011, there were almost 400 million with type 2 diabetes, and by 2030, it's expected to be approximately 550 million people in the world with diabetes. As Zoran mentioned, there is a regulatory guidance both in the U.S. and in the EU with respect to what is now required to demonstrate cardiovascular safety in drugs to be approved for type 2 diabetes. Most companies, as Zoran has mentioned, have chosen a separate cardiovascular outcomes trial, which is typically combined with efficacy trial, cardiovascular events with a meta-analysis at the time of meeting the 1.8 threshold. As most people are aware, the first application, the NDA submission of dapagliflozin, actually did not include a separate cardiovascular outcomes trial. These trials can range from 2,000 to more typically 5,000 to 10,000 subjects, or as I'll show you shortly, 16,500 subjects. Typically, their duration is 3 to 5 years at a cost of $200 to $400 million. What's just astounding about the 2008 guidance and unintended consequences is that the overall cost of executing a phase 3 program to get a drug approved for type 2 diabetes has essentially doubled because the cardiovascular outcomes trials are so large, and the efficacy trials in some programs, actually the number of patients exposed in the efficacy trials is much smaller than the number that are exposed in the cardiovascular outcomes trials. Because the cardiovascular outcomes trials are the largest and highest cost single study, they must be designed correctly. They must be designed correctly to meet the needs of the company. There is no one correct design, and this is something to be considered as companies choose their strategy. The idea that there are designs and statistical approaches to assess the cardiovascular risk with respect to the 2008 guidance does require some clarification, and this webinar and others like it are aimed at providing, if you will, experience in the field, what has been done, what could be done, and there will actually be publications in this area, and we are now aware that the Cardiac Safety Research Consortium, the CSRC, which is out of Duke University, does have listed on their web page that there will be a paper released shortly with respect to the statistical approaches to assess CV risk. And so this webinar and others should be material that prepares the listeners and participants for this paper, which will be coming out hopefully in the very near future. As we look at the cardiovascular outcomes trial landscape, let's take a look at, if you will, an assessment of the cardiovascular outcomes trials with the columns populated from clinicaltrials.gov. There are many trials listed on this page. What you can see is that the size differs, and it goes from 2,000 for the ITCA 650 study all the way up to 16,500 for the TIMI 53, the SAVOR study, with results that were recently announced. What's quite interesting here is that if you look at the size of the SAVOR study and compare it to EXAMINE, both were looking at DPP-4 inhibitors, and both actually had the same primary endpoint. They used a standard MACE definition of cardiovascular death, non-fatal MI, or non-fatal stroke. But as you actually look under the MACE primary endpoint column, you can see that many studies have used what we're calling MACE Plus or M4 as their definitions, and that the endpoint considerations are large with respect to whether you are trying to meet the demonstration of cardiovascular safety or potentially, as Zoran will get into later in his discussion, whether you actually want to try to tackle demonstrating cardiovascular efficacy in this population. The conclusion, though, from this slide really is that the selected populations vary, the size varies, and the MACE definition varies. What is quite interesting if you look at the population is that it is different for every study, essentially, in terms of at least what's listed in clinicaltrials.gov about the kinds of patients that are being selected to increase the MACE rate above the general background rate in a type 2 diabetes population. And there are slight differences in trials. If you look at Carolina and Leader, you see that one had pre-existing cardiovascular disease, the other listed concomitant CVD, another study, Rewind, established CVD, and the criteria to meet these definitions differ even within similar kinds of descriptions. The NCT trial number is listed on the right-hand column if you want to go actually evaluate those studies themselves. As we keep in mind the strategies to accelerate the NDA filing, let's do some simple math. I'm not a statistician, so I can only do simple math. But the point that Zoran will be making is that the NDA can be filed once the 1.8 threshold is met, and the 1.8 threshold being met is determined by the statistician evaluating the MACE rate of the population and calculating the number of events that will be required to meet that threshold. The number of events is a calculation of the annual event rate times subjects times the number of years that they are exposed to the drug. And therefore, acceleration of the NDA filing can be done by increasing the number of subjects. The question is big always better? And we will take some lessons from SAVOR in the next slide. Also, you could increase the annual event rate. But another question, interesting question, is the highest rate always better? In other words, if you were to define a population with such a high MACE rate, would it be possible for a drug that could potentially be cardioprotective to have the time to protect those people if their background MACE rate was so high? And so the next slide does want to introduce the concept of latency with respect to the impact of drugs that will be demonstrating cardiovascular safety or even cardiovascular efficacy. What I've done here is shown and researched and put on the slide a number of studies. Let's start at the bottom with primary prevention. The very first is the prevention of coronary heart disease with pravastatin in men with hypercholesterolemia. This was before studies had acronyms. What it looks like, though, is the two lines begin to separate at about a half year. As you look at the ASCAP study, the other primary prevention trial on the bottom right, when does the placebo arm and the statin arm separate? Again, about 1/2 year. Now let's shift to the top of the slide to secondary prevention. And as you can see on secondary prevention in the slide, the panel all the way to the left, this is the CARE trial. You see that there is not a separation between the two arms until 2.5 years. And keep in mind the 2.5 years as we talk about SAVOR and as you review other cardiovascular outcomes trials with respect to median duration of exposure. Then we look at the Scandinavian simvastatin study survival group, which is the middle panel on the top. and you see that there is a separation, probably about 1.5 years, maybe 1 year. And then on the top right, the benefit of simvastatin in heart protection study, you can see that there is a separation that appears to occur about 1.5 years into the treatment period. So let's just talk a little bit about SAVOR. This was a 16,500 patient study, DPP-4 inhibitor. Patients were exposed for a median of 2.1 years. The MACE rate appears to have been approximately 3.6%. And the question is, what was this study, if there is a latent period, which I'm trying to make a case for, that there is a latency period where even a therapy, the statins, as you know, are actually indicated for prevention of coronary disease, secondary and primary prevention. But if there is actually a latent period between when the medicines are started and when that effect first is evident, what is that latent period? And it appears that that latent period is somewhere between, I would guess, 1 to 1.5 and 3 years, depending on the study that you want to reference. And so if the SAVOR study was large, which it was, and had a median period of exposure of 2.1 years and was designed to demonstrate superiority with respect to cardiovascular outcomes, might it have potentially been at a disadvantage if there is a latent period and the MACE rate was high and the number of patients was so large that you just did not need to study them that long to see the number of events that were required. So these kinds of trade-offs are things that need to be considered when these studies are designed. So the other interesting point I wanted to bring out on the landscape slide, as we advance this slide to this is essentially the last column on the landscape slide I showed previously, is that there is a question of, does the diabetic population selected resemble the patients who would use the drug? For instance, ACS within 15 to 90 days, yes, diabetics are at high risk for coronary events, but is there perhaps over-selection or over-enrichment of the MACE rate that then studies a population that is not the typical population that would be receiving this drug? For instance, you could easily design a study in type 2 diabetics with stage 3 or stage 4 CKD. It might be very, very difficult to find a number of patients to completely enroll this study. But the point is, the 2008 guidance does allow the type 2 diabetic population to be enriched, and many sponsors, as you can see by the column here on the left, listing population, have done just that and have gone to ACS within various periods, and most likely there will be in the near future potentially sponsors going at other high-risk type 2 diabetic populations in addition to ACS, which could include such things as advanced kidney disease. So we need to keep in mind the intended population, because this has potentially implications on the label when the drug is approved. So let's talk about serving two masters. The two masters I'm talking about are the NDA safety exposure filing requirements and then the CV safety requirements, which is the 2008 document that Zoran was referring to. With respect to the diabetes-specific guidance, with respect to safety exposure, the FDA requires 2,500 with any exposure, and this is specifically in phase 3 only, as you can see from the right-hand column in the figure. There is an expectation that 1,300 to 1,500 will be exposed for greater than or equal to 12 months, and that 300 to 500 will be exposed for greater than or equal to 18 months at the time of NDA submission. And so Zoran and his company and people performing similar services can provide sponsors with a output based on advanced analytics of when the NDA could be filed based on meeting the 1.8 threshold. And that date will usually be different than when you satisfy every one of the rows here for NDA safety exposure filing requirements. And so what's interesting is that as a clinical developer, we're always working towards a critical path for filing the NDA. And if your cardiovascular outcomes trial is very large and starts very early in your program, it actually is possible that you could have the number of events that would allow you to submit the NDA upon meeting the 1.8 threshold before you actually had the requisite safety exposure to file the NDA. That is 2,500 exposed during phase 3 and 300 to 500 for greater than or equal to 18 months. And so the trade-off between these two masters is something to be considered as a phase 3 program is designed. At this point, I will gladly turn the presentation back over to Zoran and hope that some of the knowledge that I provided will be helpful as we listen to his excellent presentation from this point forward.
Thank you, Paul. And now I will get deeper into strategy and design. So when it comes to strategy, I will present two general approaches. One is to address the pre-marketing and post-marketing objectives simultaneously, and I will address that one as the default because it's the most efficient approach. And with this approach, we will just conduct one meta-analysis that includes the outcomes trial and together with efficacy trials, and that, for the sake of this presentation, this trial will start parallel with phase 3 trials. We can discuss some alternative strategies later. And pre-marketing requirement, or the 1.8 requirement, is addressed at an interim analysis for the final requirement, which in this case would be 1.3, the ultimate requirement. The second strategy would be addressing these, pre-marketing and post-marketing separately. So there are two options. There are two ways to think here. One is you have, meta-analysis of efficacy trials combined with a smaller outcomes trial for pre-marketing. You close all these trials, you file, if you're successful, and then you open another post-marketing trial separately from this previous, original meta-analysis. And, the second alternative here is maybe to try to accomplish 1.8, without an outcomes trial, but I will provide you with some numbers just to illustrate if and how that may or may not be possible. So at a high level design, just some things that need to be considered. Again, MACI trials are event-based trials. When you look at the sample size considerations, to achieve 1.8 requirement, you need 122 events if you don't have any interim analysis. And you would just add a few events to this. If you have interim analysis, you will see these numbers. But essentially, that is where you are. And 611 events to achieve the 1.3 requirement. So as Paul mentioned, there was a simple formula and can be very useful, to do some initial calculations in your mind. 711 events, what does that mean? If you can expect an annual rate of 1%, that means that you need 61,000 patient years to accomplish the 1.3. Or if you have 2%, then you would need about 30,000 patient years to accomplish the 1.3 requirement. That means 30,000 patient years, about 3,000 patients for 10 years. Or 6,000 patients for 5 years. So you can, in your mind, start playing with some numbers. They're going to be just roughly what you can expect. But this is where you are. This is what this guidance means. And this is assuming that hazard ratio is equal to 1, which means that the treatment is equal, absolutely equal to the control when it comes to these events. Other things that need to be considered, these are rare events, and the consequence is really what I just explained, and that is, that you need to wait for a long time to accrue these events. Also, that means that you can learn very little from your early trials. These events do not accrue. They don't accrue very fast, so you can learn very little from early trials. That means that there is a lot of uncertainty in terms of what you know a little bit about your drug, but you don't have data, when you start planning these trials to really support your assumptions. So de-risking is highly recommended here for this reason, and for the reason that these trials are going to cost you, hundreds of millions of dollars. And finally, the thing that we mentioned and that the guidance specifically mentions is meta-analysis involved. So you need to be combining data from a number of different trials, and then you're running the analysis on this combined database, the data that includes coming from many different trials. So this was at the high level. So strategy number one. So again, this is strategy number one from the slide that I've shown before. Address objectives simultaneously. So let's make some assumptions. There are many ways, as Paul already mentioned, that you can go. So we want to address 1.8 and 1.3 simultaneously. So that means we're going to test sequentially. Once we accomplish 1.8, then we can go for 1.3. And I have a chart that illustrates this. Meta-analysis, so we are going to incorporate meta-analysis here. So we are going to combine data from phase 2b, 3, and outcomes trial. Most data will come from the outcomes trial. You will see that. So 1.8, we can go many different ways. We will, in this example, just assume a design that has one interim analysis and a final analysis. Now, an important thing to consider for the first interim analysis, this interim analysis cannot be scheduled before you finish your efficacy trials, because you need your efficacy trials for filing. Not only that, but there is no point really in scheduling this interim analysis before you accomplish the other safety requirement that Paul illustrated in his last slide. So that is one thing to consider, and careful planning is important here. So second interim analysis of this meta-analysis is going to be serving for the final analysis for 1.8. And really just to come up with a design for 1.3, we're also going to have an additional interim analysis for 1.3 and then the final analysis for 1.3. What does that look like? Well, I have another slide before I show you the chart. I want to show you the impact of interim analysis and various strategies. So as I said, we are going to schedule an interim analysis relatively late. Here I'm scheduling it at 75% of information that we have, for the final analysis for 1.8 requirement. And this is really just for your illustration to see how these things work. Then we have two types of what we call spending functions. And for people who are not statisticians, they don't need to know everything about them. But this O'Brien-Fleming is a less aggressive boundary, and the POCOC one is going more aggressively at the interim analysis, but it's spending more alpha. So how do these interim analyses help us? Well, with 1.8 requirement, if we have no interim analysis, we are going to have final analysis at 122 events, but the expected number of events is also 122 because we have no options to stop early. If we go with this O'Brien-Fleming, which is not a very aggressive function, then we have approximately 70% chance to stop at the first interim analysis, which is at 75% of events. So we have a good chance to stop the trial early. Because we have to adjust our alpha, the maximum number of events that we may need is 124. However, the expected number of events is 103, which is much better than what you would have without interim analysis. And in this case, POCOC function gives you a very good chance to stop early at 82%, but because of alpha adjustment, the final analysis comes after 135 events. And then the expected number of events would be around approximately 107. So in this case, O'Brien-Fleming would be preferable over strategies, either over strategy with no interim analysis or over the POCOC strategy. O'Brien-Fleming is not always going to perform better than POCOC strategy. If you have a look that is earlier than 75%, then it's possible that POCOC would perform better in terms of the expected number of events. O'Brien-Fleming would always be better in terms of the maximum number of events. So these are really just some things to consider. Similar situation with 1.3, except that the difference in expected number of events between designs with interim analysis and no interim analysis would be even greater. So de-risking is of a great help here with these trials. This is now how would this design chart where you would go simultaneously after both objectives look like. And I will try to use some of these WebEx tools. Hopefully they work. So as I said, the first interim analysis, the first analysis would be the first interim analysis for 1.8, and would be the very first interim analysis for 1.3. For 1.8, it would come at 75% of data. That's how I schedule it. At the same time, as the numbers play out, it would be approximately 15% of this number for 1.3. Then you would have the second interim analysis where you test again for 1.8 and have option to go down to 1.3 if you achieve 1.8. As I said, 1.3 is much more difficult to achieve, so there is no even point in looking at 1.3 unless you achieve 1.8. And also for the sake of protecting alpha for, statisticians on this call, obviously you need to go sequentially anyway. Now, 1.8 requirement you must accomplish at this because you don't have any other looks at for 1.8 after this point. And 1.8 is required for your submission. So if you don't achieve 1.8 at this point here, that's it for your development program. There is no more. If you do achieve that, then you can go and select in this interim analysis. I use 50% and 100% really just for illustration purposes. There is no good reason to just always go with something like that. You need to do thorough simulations of a kind that I will be describing during this presentation. Now I will go to the next slide. And this type of table looks busy, but I think it's extremely important for planning. Now, we are designing this trial thinking or assuming that the true hazard ratio is 1, the two treatments are identical. But in reality, they can be different. Your drug can be doing better than the control or possibly even worse than the control. There are a number of things that you can see in from this chart that can help you with many things. For example, with 1.3, obviously, we said we are going to go sequentially, but we have very little chance to achieve this prior to much later in the study. Nevertheless, there isn't much to lose when it comes to a number of events, so you may as well keep the design as is. You can look at the spacing of interim analysis. For example, you're not sure what your true efficacy may be, so with 0.8, you have very good chance to stop early. However, if the true efficacy is 1.0, you may be forced to go all the way to 613 events, and that's a lot. So just looking at this, it may be wise to incorporate even more interim analysis, in this period between, submission and the final analysis for 1.3. Really just for the sake of de-risking and optimizing your design. You can also compare Pocock and O'Brien's spending functions, see which ones perform better. A chart like this can be very useful for many different reasons. But there are a couple other things that you may look at. For example, if the true efficacy is 0.8 or 0.9 true hazard ratio, your power is really great here, for accomplishing all of these objectives. You may be compelled to consider showing superiority, not just non-inferiority as the guidance requires, but superiority as well. On the other hand, if you're in the range of 1.1, and I didn't show 1.2, the power drops very fast, and particularly for 1.3 requirement. So you may want to look into some other ways of protecting, your trial and your development program. So let's address some of these. The first one is, well, what if the hazard ratio is actually greater than 1? So we said, well, 1.8, we have pretty good power if hazard ratio is 1, but that power drops, very sharply if the hazard ratio is greater than 1. And 1.8, your development program depends on this. So you may want to incorporate another layer, of safety, de-risk a little more. So you can give yourself an option to increase the sample size to provide yourself with an even more power, and you can assess whether you need more power or not at this first interim analysis here. And depending on the trend that you observe, you either stay with this, as a final analysis, if the hazard ratio is assumed. But if the hazard ratio is not such, then you may want to increase the sample size and have yet another look somewhere after, 124 events, just to give yourself an even better chance to accomplish what is required by this guidance. And with this type of strategy, we keep on enrolling to accomplish 1.3 anyway. So this sample size reassessment, will be just, coming in terms of additional number of events. We're enrolling patients. We keep on enrolling patients anyway. We can do the similar thing for 1.3, you know, if it turns out that the true hazard ratio is greater than 1, but then you would have to consider enrolling more patients and accruing more events than 613. Then the next question, Then the next question comes, well, but what if we on the other side of hazard ratios on the other side, it looks more promising. Should we consider superiority? Well, the first reaction may be why not. From the design standpoint, you just may have a little, you have more looks, and you go sequentially. And you accomplish 1.1 first, and then you go ahead and if you accomplish 1.3, you continue on, and then you can go down to superiority. And most likely you're going to accomplish 1.8 first, and then 1.3, and then superiority. So the chart is going to look something like this. But then the question is, when is superiority worth considering? So first of all, you need to look at some numbers. So this is what you would need in terms of numbers for superiority, assuming no interim analysis, as you have seen, incorporation of interim analysis, particularly with O'Brien-Fleming function, doesn't add much here. But if the true hazard ratio is 0.75, then you need about 508 events. 0.8, 845, 0.85, about 1,600 events. So let's do a quick calculation. Let's assume again that MACE rate is 2%. So 1,600 events, that is about 80,000 800,000 patient years, which is about 8,000 patients for 10 years or so. That may be excessive already. So being at 1,600, if you anticipate that this is where your efficacy is, it's questionable. It's questionable if you should go for something like this. This should pretty much be out of the consideration. Now, these numbers here don't look too bad, but if your true hazard ratio is 0.75 or 0.8, you're probably going to accomplish your 1.3 much earlier than 600. So this is an additional, a large investment regardless. So then you should be asking yourself, from clinical commercial standpoint, is it worth or not? And I think this is a good time to switch to Paul for at least a couple of minutes to provide us with some insights on that.
Sure.
Ben,
I
would appreciate. Yeah.
I just have a few observations here. The desire to demonstrate superiority clearly is only a path that should be pursued if it is felt that the hazard ratio of the experimental agent is less than 1, and we know that that there are classes of drugs where this is believed to be true, and the idea here is almost along the lines of if you have lemons, turning them into lemonade. If the burden on a company is to demonstrate cardiovascular safety, and the burden on all companies is to demonstrate cardiovascular safety, we know the reason for that. We know what analysis led to this 2008 guidance. But now that there are mechanisms of action that potentially have hazard ratios less than 1, the question is what is the value if we have to demonstrate cardiovascular safety of actually extending the study longer to be able to demonstrate cardiovascular efficacy. And it is published that some of the DPP-4 inhibitors have designed their trials with that in mind, and the commercial value of having data and label or an indication to reduce cardiovascular events in high-risk patients with type 2 diabetes would be a differentiated label, and the companies are then able to assess a value to that differentiated label. And that's why the number of events that are recorded in your right-hand column that looks so astounding, and we know that they are very costly to attain, they do have a commercial value. And so obviously a drug needs to be safe for type 2 diabetes, but if there is a potential for that drug to demonstrate cardiovascular protection, then that is an additional factor that's taken into account when calculations on NPV are made based on the target product profile. And so we have seen studies that have attempted to address this, and we will continue to see studies report out that are looking at this robust, if you will, label claim or indication. So, Zoran, I think I'll turn it back over to you now. Thank you.
Thank you very much, Paul, and that was very insightful. And as you said, I think a great point. Rather than just guessing what a number of events may mean, we can calculate the cost and the expected length of development or additional length of development, and potential additional revenues, and really based our decisions on that. Thank you very much. So then, I think this is the final slide of this section. There are just a couple other things to consider. One is the population definition. As we know, usually for superiority trials, the ITT population is required. With non-inferiority trials, you know that regulatory agencies usually require patients as treated, and the reason for that is that with ITT population there is a tendency of efficacy or results going towards zero, and when you have superiority trial, that makes it more conservative. However, with non-inferiority trials, the ITT population may actually, analysis on ITT population may be less conservative. So in all likelihood, the FDA is going to require you to run analysis on both ITT and treated. Futility assessments certainly to be considered, with such large trials. Now with 1.8, with the futility assessments, you're just saying, well, I just don't believe that this drug is going to work, because you absolutely need that one. However, for superiority, it's probably worth, you expect an extremely large trial, so it's probably worth incorporating futility analysis, really just to de-risk your program. And now filing and preserving the outcomes trial integrity. So for the filing purposes, obviously all efficacy trials need to be unblinded, so they all have to be finalized prior to submission. But for the first strategy that I presented, for this strategy as presented, the outcomes trial has to be ongoing, and the current standing with the FDA is that the sponsor could be allowed to keep this trial blinded. There's some ongoing discussion, but that's certainly something that every company should check before taking this kind of approach. As I will show in my upcoming slide, this approach is a lot more efficient, and I think everybody understands that. And for that reason, there would be really a great advantage of just keeping this trial ongoing. But this issue is something that has to be kept in mind. Strategy number 2, I will go over this much faster. So the strategy number 2 is similar. You're addressing objectives separately, and then you go for your 1.8 requirement, and then you file then, and for 1.3 requirement you start from the beginning. Now here, as I said, you can either have a smaller outcomes trial and then initiate another one here, and you will probably need a smaller one here, or you can try just to accomplish this with meta-analysis, and I will illustrate that. So, what advantages and disadvantages? Well, obviously, with this strategy, you lose all the events that you collected to address the P-marketing strategy, and that's around 120 events. That's not little by any mean. Then you have all that white space. You have filing, you have preparation for the new trial, reinitiation of enrollment, and things of that kind. Overall, obviously, increased costs, delays in accomplishing a number of milestones, and certainly delays in achieving post-marketing goals. Advantages: this would be easier to design and implement for some reasons that have been discussed. And also, I said previously, when you're planning these trials, you have very little knowledge about what you expect. Well, one advantage is, going into post-marketing, now you have a robust database, that you, from your pre-marketing trials that you can use for planning of the post-marketing trial. A meta-analysis only, obviously, one would wish that that pre-marketing 122 events can be accomplished with from efficacy trials only. But there are a few problems. They're smaller in size. The population in these trials is not enriched, for cardio. It's not high-risk, cardiovascular population, so you can expect low event rate. And I will provide you with the numbers in a couple slides down. And you can consider extending efficacy studies to gather some additional to collect more MACE events, but you still may not have sufficient power. And as I said, you may be delaying your approval by doing so. So it's some issues there that one needs to consider very strongly before taking this kind of approach. I will go into the final section of this presentation, and as I said, so far everything that has been presented here, has been presented in terms of events, and we did show some formulas about how to, get some idea of what these events mean. But exactly how does this translate into number of patients and development times? You want to have larger studies that go longer? Would you like to enrich the population or not to enrich? How can you project, timelines of interim and final analysis? Submission timelines, planning, for final and interim analysis. Considering the impact of combining, meta-analysis, combining efficacy trials with outcomes trials. You need to answer all these questions once you already have your strategy in terms of events defined. So for this, it's absolutely necessary to have tools for predicting enrollment and accrual events so that you can adjust all these, so that you can compare different strategies. And also, as you can see in the bullet points right before the last, monitoring the development, monitoring the accrual of events. Is everything going as planned, or do you need to have some corrective actions? Do you need to change something in the way that the trial is going? So I will present you some simulation overview. So the first thing that you need for these simulations is really what is coming from the previous part of this presentation. You need the sample size in terms of number of events. You need events model and parameters, and you need to define your milestones, things like, interim analysis, your filing times, things of that kind. Then you need to get the information on enrollment parameters. You have to go as detailed as regional data, country, data at country level, site data. You look at the enrollment rates and dropout rates, from different, regions and sites. And then you look at the site activation plan. Once you have this in place, then you simulate scenarios. And so you simulate the site activation. Once you have that, the next step is you simulate patient enrollment. In these simulations, once patients are enrolled, they can start accruing events. So you need some assumptions at what speed these events are accrued, and also dropouts, which is also an important part. In consideration of final sample size in terms of number of patients. Once you have that, you get your simulation results, and with programs like this, and actually you will see in our example as well, not everything is going to fit, not everything is going to match and fall in place as desired. So almost certainly, once you get the first set of outputs, you will have to. Oh, I'm sorry. You will have to do this. Go again and run several iterations till you get closer to exactly what matches your strategies, and provides you with the most efficient, more realistic and most efficient development. I will not go into details here, but these are really some ideas about, some background about these enrollment and event simulation models. First, you will start with some prior information for both of these. So you have some prior information for enrollment, and you have some prior information for event accrual. But then as the study starts, then you get some data from your own study. So having Bayesian updates, the simplest way to explain this is you're not relying on the historical information alone, and you're not relying on the information from your study, but you're really having some kind of weighted, estimate of combined, information from history and what you're observing in your own trial. For enrollment models, a typical assumption, a gamma model, and non-statisticians really don't need to worry about this. Gamma distribution seems to be fitting enrollment rates very well, and for events, a very traditional approach is to assume exponential function, exponential accrual, and in this case you would go, for update, with piecewise exponential model. These are our case study assumptions for the outcomes trial, and we also just drafted some kind of plan for efficacy studies so that we can simulate accrual of the rates from these trials. I don't know, Paul, if you would like to say anything about the rates that have been assumed of 4% and 0.6%. Respectively for these two types of trials?
Yes. Just as you can tell by the NDA filing being based on the number of events that occur, calculations with respect to the MACE rate for the population or assessment of the MACE rate for the population that's being enrolled are critical. And there is a literature which obviously lags behind real-time in many ways, and there is recent experience. If you look at the SAVOR study, as I mentioned before, the MACE rate in this population appeared to be close to 4%, 3.6, 3.7% or so. If you look at EXAMINE, it was higher, but it was an ACS population. Typically, there are ranges that center around 4% for the high-risk population as it's defined, and as you saw from the table I presented earlier, the high-risk population is defined in various ways. And as you now look at the non-high-risk MACE rate, there is a lot of variability. Should the typical diabetic that is not at elevated risk, as in ACS event or other factors which increase risk, be considered to have an elevated risk? Most people would agree yes. The MACE rate in that population of between 0.4 and 0.7% has been seen or used by some estimates. And as you are able to gather from the size and the length of the efficacy trials, perhaps the precision around the MACE rate prediction for these trials is not all that critical, because there are so few subjects in these trials, and they will be contributing very little to the overall rate. And as you can see in this slide that Zoran is posting, you have essentially trials that have, in this entire program, which is shown as a 4-study program, which is probably quite small for a type 2 diabetes efficacy program, you see that you have less than 1,800 people that are in the non-cardiovascular outcomes trial. And so the estimates in these populations are, on the one hand, important so that you can prepare for the meta-analysis or prepare your expectations, but they contribute very little because the length of the trials are so small. The other thing to keep in mind is that standard of care in diabetes and all fields of medicine continuously evolves, and typically the standard of care which led to a MACE rate prediction that is started or that is used for statistical assumptions at the beginning of a trial is no longer accurate as the cardiovascular outcomes trial progresses, because standard of care has advanced and actually the MACE rate in that population is going down. Zoran, I think I hit most of the high points there, and I'll stop for now.
Yes, thank you. And just one thing for everyone, just please remember this last point. We are assuming the trial starts October 2013. So given all this, let's see what do we get. So we set, 6 different milestones. I think these milestones from 3 to 6 are most relevant here. But, we have some milestones for, let's say, enrolling 6,000 patients, 12,000 patients, and then we have milestones for events. And these milestones are the ones that are important for our development, obviously. So we said October 2013. And we set some goals, and then December 31st, well, that's only 15 months after October 2013, and even this February, 28 February, that's about 18 months. So we are not going to have enough time to accomplish the requirement for the exposure. As you remember, we need several hundred patients who have been exposed for at least 18 months. So at the first step, with developing this strategy, we came into something that is going to require us to actually make some additional iterations, and go back to our strategy and correct it a little bit. Aside from that, you can see from this chart what is the probability of achieving. So the output that we have with these type of tools, simulations, the output is really probabilistic, and you will see on the next slide, you will see what it looks like. But let's say for 600,000 patients enrolled, we have about 13% chance to accomplish that by the 31st of December. So rather than saying, okay, we are going to accomplish that, by a certain day, we are assigning some probability based on our simulations. What is the chance that we are going to achieve that? Now, if we combine that with phase 2 and 3 efficacy, so this is only from the outcomes trial. If we combine that with efficacy trials, then this goes up sharply. Now, the enrollment, as I said, may not be that important, and after all, these patients in efficacy trials are not followed for as long as patients in outcomes trials. So that may not help us that much. But here we have 60% chance of achieving this one. And, efficacy trials are going to add, to elevate this up to 77%. So they're contributing. As little as they are, they would be contributing here. Something to keep in mind. Once we see this, the next step, we want to go into a little more detail and see what this chart really looks like. Oh, I'm sorry. This is yet another specific here. Here we are looking into exactly how many events do we get from efficacy trials. So by 31st of December or 28th of February, all that we get is 6 events from these trials. So it's a rather lean development program that we had, and we assumed a very small MACE rate for efficacy trials. However, if we were planning to achieve the goal of 122 events from efficacy trials, I think it's fair to say that is extremely, extremely unlikely. We cannot get anywhere close to that. But we do get some contribution to the number of events, for our database. And now what we need from the outcomes trial, and this is important for the planning of the outcomes trial, is 87 instead of 93 events. But there is another little problem here. Well, as it turns out, we keep on accruing these, events from efficacy trials even past the point of, the latest point of submission. What does that mean? Well, the way that we position these efficacy trials and the proposed length of trials goes beyond the point where we where we expected to have submission. And as I said, these trials have to be unblinded by that time. So obviously we need to go back to drawing board and move some things around a little bit for yet another reason here. This is what the output from this tool would look like. So as I said, what you get is a distributional output. So we said it was about 13% chance, so we're about here, that we are going to accomplish our goal of 6,000, given what we have assumed. So that's something that we know. We can also say, well, for how many patients can we accrue with 50% chance? What is the average expectation? And that's around here. So we can say, okay, well, we have 50% chance of, accruing about 5,700 patients. Again, this type of output gives you a little more insight about what is going on and what can you change. Also, you have some areas that are rather flat, and some areas where you can, you have a steeper slope, which means more improvement along the line. A similar graph you can develop for accruing events. And so what did we say with this is for 93 events. We had close to 60% chance. But if you get 6 events from efficacy trials, then all that you needed from the outcomes trial is 87. That is why we were at about 77%. And this is the part of this curve that is very steep. So that's why you get almost 20% additional chance of achieving this just by adding 6 events from efficacy trials. So these are all little details that the planner can look into. And then you have predictions. So this is what you know in terms of number of what you can expect in terms of enrollment, and you have put some confidence intervals. So if you're 95% confident that you're going to be in this range, for 12,000 patients, with 95% chance that you're going to accomplish that between these 2 dates. Now, as you start enrolling, the trend may go, let's say the trend goes like this, a little bit under here. So then you can, this whole curve changes, but not only does the curve change, but up to this point, if you're looking here, you don't have confidence interval, now you have a straight line. So now whatever happens from this point on is slightly narrower. So the more information that you gather from your study, the narrower this 95% confidence interval is, and the more you know about your targets, and the more you know about how you should, if and how you should adjust your strategy when it comes to enrollment, when it comes to the population that is enrolled, and whatever else may be of relevance. So just a few points about the simulations discussion. So given the assumption that we use, it would be extremely unlikely that pre-marketing requirements can be achieved without an outcomes trial. So whether you go with strategy 1 or 2, you probably, your chances of achieving approval without an outcomes trial are very slim. Since a small number of events that can be expected from efficacy trials, one can consider using events only from the outcomes trial, then you have more homogeneous population. However, there are a couple of things that one needs to consider here. Number one, actually the FDA guidance reads as if it should be assumed that you should be running the meta-analysis. Whether you have to or not, you can discuss with them. But also do not remember this second safety requirement for exposure and strategic positioning of these efficacy trials can actually help you and make your chances of achieving approval earlier, can make them better than if you go with outcomes trial only. Similar simulations can be implemented to assess the impact of enrichment, for example. If it should be a no-brainer. If you enrich population, you can accrue events faster. You should probably go with that, but there may be an impact on enrollment and many other things. So really just running, some options, a few simulations just to see what you can expect under different scenarios certainly wouldn't hurt. And most importantly, these simulations are necessary to predict timing of interim and final analysis, not only at the beginning of the program, but also as you go and as you're learning more about enrollment rates, real enrollment rates, and real, event rates, updating and changing the strategy, on the fly. As if all these considerations that satisfy just the MACI guidance were not complicated enough, as Paul mentioned, we do have this additional requirement that does bring up some additional complications and considerations. Paul, it may be a good time that I switch back to you again and go over this a little bit.
Thank you, Zoran. Without actually reading the slide, the points I want to bring out are really just stressing what has been brought up previously in the conversation, and that is that, as Zoran and I have discussed, we always have to keep in mind the safety requirements for filing any NCE, which are different than the actual cardiovascular safety-specific requirements. One of the interesting things is that as we have more data on the annual MACE rate of high-risk populations, we now perhaps have the freedom, if we're designing a cardiovascular outcomes trial to demonstrate cardiovascular safety, not cardiovascular efficacy, to start it. It doesn't have to be necessarily the first study to start, and that's demonstrated by the modeling advanced analytics that allows the prediction of when the safety exposure will be met with respect to 2,500 any exposure and the number of MACE events, let's just say 122, to satisfy the 1.8 threshold. And so the clinical operations groups become even more important than they used to be, and they used to be one of the most important groups for actually delivering a trial that clinical development would be designing. So now the actual timing of the efficacy trials, the timing of the largest efficacy trials with respect to the cardiovascular outcomes trial become critical. And then that has implications, if you will, for actually what is the short-term database lock of the efficacy trials, and how long will the long-term extensions be. And there are many trade-offs to be made when you are considering the earliest possible NDA filing date. And as we gather more knowledge, the expertise of people who've had experience in designing these cardiovascular outcomes trials and being able to use the interim analyses to make decisions about when to file or when to delay filing, perhaps based on the outcome of these decisions, will be critical. And so this slide just lays out that there are a lot of choices to be made. There is no one right choice for any company, and there are many factors to be considered when deciding how large to make the cardiovascular outcomes trial, how high a MACE rate to have in the population based on your inclusion-exclusion criteria, and how long to plan to have that trial be executed for, which, as I mentioned earlier, is critical if you are actually looking at cardiovascular efficacy. Then you may want to consider a slightly longer trial to allow for latency period considerations. Zoran, I think I'll stop there. Thanks.
Thank you. And this would be, I think, the very last slide of this presentation, so really just some summary points. So large outcome trials are causing large increases in cost and risk in drug development in diabetes and actually a number of other indications. So the first planning step should be to assess which development strategies are reasonable and or more efficient in terms of number of events. Reasonable based on what you know. Should you go for superiority, for example, or not? And then for most efficient, certainly, how frequently do you want to have interim analysis, what kind of spending function to use and things of that kind. Once that is determined, then the next step is to run simulations to determine what it means in terms of number of patients and length of development. And we started with an assumption, I think it was 10, 12,000 patients for our, but in the real life, we probably would have considered several different sample sizes. And then we would get, that would have an impact on the length of development, time of approval, and then, based on simulations, we would see which ones suit as the best. Similarly, decisions on, whether to enrich or not should be made, whether to combine, your outcomes trial with efficacy trials. All things of that kind can be really best addressed by simulations, and you can use them to compare various scenarios and really decide what works best for your development. As I mentioned, and hopefully illustrated, obviously I would need a lot more time to illustrate, how you would go for a number of iterations, but usually a number of iterations are needed to determine the best strategy for number of trials, sample sizes, sequences of trials, timing of interim analysis, many questions associated with these trials. Once the strategy and design are finalized, then simulations are necessary for predicting and, more importantly, repredicting, the timing of the analysis, just making sure that things are developing as planned, and if not, make corrections, make adjustments. And I think that's all that we had to say for this presentation. Thank you, Paul, and thank you to everybody who has attended this webinar.
Yes, and hello everybody. My name is Mike White, and I lead the marketing efforts here at CITEL, and I'll be acting as a moderator, if you will, to pose your questions to Zoran and to Paul. I first wanted to remind everyone that today's slides are available for PDF download simply by going to citel.com, our homepage, cytel.com, and on the right side of the homepage you'll see a link for today's event, and then you follow that and download the PDF of today's slides. I did want to right away get into your questions, but we'll also start with a question that has been asked oftentimes in our discussions regarding CV outcome trials and the guidance issued by the FDA. Zoran, you made reference to the specific guidance here for diabetes research that was, I believe, issued in 2008, was that?
Right.
What about other related areas, therapeutic areas? Are there other guidance that may be applied perhaps to obesity, anemia, or related blood disorders and other areas where you would receive outcomes studies are recommended?
Yeah, thank you for asking that question. Between Paul and I, I'm sure we can address that. I'm personally not aware of a formal guidance for any other indications. I may not be up to date. I know that EMA, in discussion with some regulatory people, that EMA did not have a guidance for diabetes, but in general has agreed that the FDA requirement is reasonable and along the lines of what they would expect to see. Obesity, I'm not familiar with the guidance. I know that some companies receive the requirement that's a little more relaxed than for diabetes, I think along the lines of 1.9 and 1.4 rather than 1.8 and 1.3. And I've heard from many of my other colleagues from working on other indications that requests either in terms of formal numbers or requests of a kind of CV studies that will be discussed with the sponsor further have been made. So that is as much as I can tell. I don't know if Paul can add anything to that.
No, Zoran, you summarized it quite well, that the Diabetes 2008 CV safety guidance is being used, if you will, as a anchor for other therapeutic areas to set their expectations on what is required for CV safety, and you were right on target with obesity. Thanks.
And Paul, while we have you on the line, you stressed earlier that that MACE interpretation differs from program to program, sponsor to sponsor. Can you elaborate some on the different considerations for use of MACE or MACE Plus as endpoints?
Sure. What's interesting is that the the 2008 guidance document did lay things out in a way that appeared to give some freedom. The guidance document essentially stated that the events of interest should include cardiovascular mortality, myocardial infarction, stroke, but that they also could include hospitalization for acute coronary syndrome, urgent revascularization procedures, and as I said, quote, possibly other endpoints. It's the possibly other endpoints as well as the endpoints in addition to cardiovascular mortality, myocardial infarction, and stroke that have led some sponsors to define the endpoints in terms of MACE plus, which is the typical 3 MACE plus something else, which typically is hospitalization for ACS, urgent revascularization procedures, or unstable angina. And there actually is some advantage to considering exploring with the regulatory authorities, whether different definitions can be used. The sense is that the MACE plus, if you included revascularization in your MACE plus definition and your trial was successful, then a prescribing physician might have a reason, a more compelling reason to use the medication. And there are, of course, issues sometimes with defining whether a MACE plus event has occurred. For instance, is unstable angina only defined in terms of hospitalization, or does it actually require validation with an imaging procedure? And there are many considerations to be taken. And then probably, I bet there will be a question about this from the attendees. There are issues now potentially with if a MACE plus, if you're looking at a MACE plus definition, does that limit the kind of analysis that can be done, and also, or what are the implications, if you will, for keeping the study blinded, and do the regulatory authorities have opinions there? So I think I'll stop because I realize we have about 6 minutes left, and want to respond to questions that will probably be directed more to Zoran.
Yes, thank you, Paul. Carlos Perdomo asks, do the presenters have any references, specific references, and I guess this would be posed to Zoran to begin with, any specific references to support your presentation, especially the simulations for predicting and repredicting the time of analyses, inter-analyses?
Okay, so the answer is yes. For the enrollment, there is a paper, I think 2007, Statistics in Medicine, Anisimov and Fyodorov, using gamma distribution, and that's actually the method that the tool that I described has used. And for event prediction, there is a paper also by Anisimov, I think 2013, I think also Stats in Medicine. Now, this tool has done something slightly different from that. And also there are existing tools. I don't know how many companies may have them. We certainly have a tool. It's of a great importance for planning interim analysis. So yeah, there is selected literature. When it comes to events themselves, I think the assumption of exponential accrual, that's something that has been around for decades now. So that there are many existing publications that would refer to that.
And Paul, going back to, we had mentioned other therapeutic areas, and I recall that you mentioned that there's something to be learned from renal disease contributing to MACE rate. Can you address that, please?
Sure. There was an interesting CME exercise a couple of years ago that referenced a paper by Levy in Kidney International in 2011, and this paper has a wonderful table that lists the mortality and kidney outcome risks based on GFR on one axis and albuminuria on the other axis. And although it doesn't break down to the typical MACE endpoint, it just talks about all-cause mortality, cardiovascular mortality, and kidney failure. It does give a range, if you will, of relative risks based on GFR stages going from what they characterize as G1 all the way to G5, which is kidney failure, versus albuminuria stages, which range from A1 to A3. And so it's possible with this table and references in the bibliography of this paper to understand how profound an effect on MACE rate kidney disease can have, and that may actually be a kind of population that perhaps could be considered as these trials are designed.
Thank you, Paul. Zoran, you showed in your simulation flow slide, you mentioned accounting for dropouts. And Carol Connell asked specifically, how does one handle subjects who discontinue treatment? Are they still followed for events afterward? And if so, for how long?
So, yes, so as I explained, they will still be followed up. And as explained, then two analyses are probably going to be requested. The ITT analysis, which will include all the events while the patient was followed up, and maybe while treated analysis. So both pieces of information would be very important. A little bit more important, almost, piece of information would be patients who dropped out of study, because those patients are going to affect, your predicted times of analysis. Particularly if there's a substantial number of patients who are no longer followed up. Then you have a smaller pool, from which you can expect events. So you would have to account for that.
Yeah, Zoran, maybe I can add to that.
Mm-hmm. Yes.
Sure. So it's so hard to remember what's in public domain and what isn't. I would bring the questioner, the person who asked the question, you might want to refer to a paper from the American Heart Journal. First author is Ben Skorica from 2011, volume 162, number 5, where I don't actually have the page number, but there actually is a section that is devoted to study retention and minimizing missing data, where loss to follow-up is discussed. And so this would be a very good place to start.
And as a follow-up to Carol's question, Maureen Silverman asked, do we need to perform additional safety assessments once a subject who discontinued treatment completed the end of treatment visit, other than a follow-up for MACE? Paul, maybe that's a question you could address.
Yes, it's the article I was referring to actually goes into both loss to follow-up and withdrawal from therapy, and the strategy that's employed in successful studies of this nature to have very low loss to follow-up, because as we know from non-diabetes drugs that had issues getting approved, a company cannot afford to have loss to follow-up if the regulatory authorities will assume that people lost to follow-up may have had a MACE event. And so the additional question I think is addressed quite well in the article that I referenced.
And Paul, while we have you on the line here, Chandra Wark asked, what percentage of MACE can typically be determined and or confirmed by imaging?
Yes, that's a very interesting question. I'd actually like to turn it around a little bit if I could. These trials are so large that typically they are not run only in countries where imaging exists. And so if you are asking that question, you may want to consider the impact on your program if you require imaging. And there may be benefit to allowing imaging if the technology exists, but be wary of painting the program into a corner.
Thank you, Paul. Jason, I believe it's Zilanka, asks, was the 4% event rate in your case study hypothetical, or how did you reach that? Was it reached via modeling?
Well, I guess both of us can respond to this. Well, 4% is, I think Paul has explained where this background information was found from, certainly from some historical information. It's not related to modeling at all, but it's something that we plug into models. We plug that as an assumption when we run our simulations and do our predictions. Now, if you remember, as I said, once the study is initiated, you have a chance to see whether your population is reflective of and the event rate is reflective of what you expected before, and then you can adjust. So, Paul, I don't know if there's a need to respond one more time whether this was hypothetical or not. I think you explained it.
So actually, I think that the slight twist on the answer, I agree with everything you said, Zoran, is that 4% is what you see in the literature. But then the question is, how do you know that your inclusion-exclusion criteria are going to get a annual MACE rate of around 4%? And literature is one source, but typically literature does not provide the inclusion-exclusion criteria to the level of detail that will allow you to probably feel comfortable or defend to your management the choices of the inclusion-exclusion criteria. And so in that case, there is modeling available of large populations using NHANES database, Archimedes, and other databases can use these resources and provide if you will, a benchmark MACE rate, and then CITEL, using either a modeled MACE rate or a MACE rate that someone says, this is my MACE rate line in the sand, can use those data to generate the wonderful analyses that Zoran showed in this presentation.
Thank you, Paul. And here's a question from definitely for a statistician coming from Khalil Saikali. Zoran, are there any regards to adjusting the type 1 family-wise error rate in these interim analyses?
Yes, so absolutely everything has to be controlled at two-sided alpha 0.05. So for each one of these requirements, 1.8, 1.3, and we can say and superiority as well, if that is what you want to go after, you're controlling, for that particular one, you're controlling alpha by the spending function. And the overall alpha is then controlled by sequential testing. So you don't go into 1.3 unless you have already achieved 1.8, and you don't go to superiority unless you have already achieved 1.3. So you almost have a grid, that controls the overall alpha for the whole program.
Yes, and I think we're going to finish today with a last question that I'll assume would be we could start with Paul on. And Paul, Carlos Padoma asks, how is vital status information from public databases used for subjects that are lost to follow-up?
Oh, that's fascinating. These lost to follow-up strategies essentially engage specialized services that actually, well, first of all, informed consent can be drafted so that there is the ability to reach out. But if that fails, there is the ability to use public databases, in a way that is totally consistent with local requirements for privacy. So I know that the local databases are used. I'm not an operations person. I don't have that kind of expertise, but I do know that there are CROs that have done this for large sponsors trials, and if you look at that table that was presented initially listing the landscape of cardiovascular outcomes trials, and then you go to clinicaltrials.gov, you may be able to trace back, what are the CROs that are involved, and so these, if you will, centers of expertise within operations are required to not have loss to follow-up. I'm sorry I couldn't be more specific than that.
And since we're over time, I will pose this very last question to both Paul and to Zoran. At the same time, on behalf of Paul and Zoran, we'd like to thank you for participating. It's evidence of the type of audience that we have by the questions that were being posed, and we're very sorry that we don't have the time to get to everyone's question. But again, we really appreciate your interest. So our last question for today from Lan Nguyen, I believe. Can the presenters discuss the considerations for placebo-controlled studies in primary versus secondary CV population and the impact of background of the standard of care?
Paul, I think I would pass this on to you. Is that fine?
Sure. If I understand the question, well, these the diabetes studies we've talked about in general, with the exception of one cardiovascular outcomes trial that's listed on clinicaltrials.gov, I believe that all the ones that are on that table are versus placebo. And the interesting thing is that for cardiovascular outcomes trial, we have to kind of shift our mind, those of us who are involved in diabetes clinical development, because when we think of placebo-controlled trial, we think of anti-diabetic medicine in one arm and placebo in the other, and you can't adjust the anti-diabetic medicine. The cardiovascular outcomes trials, because they're of such long duration, and because of the requirements or guidance or actually correspondence from regulatory authorities, they really do insist, and this is consistent with, I think, good ethical research, that the anti-diabetic care be standard of care, that the antihypertensive care be standard of care. And so when we say placebo-controlled, they are placebo-controlled, but anything can be added with respect to anti-diabetic therapy to that placebo arm. Usually, with the exception of it cannot be the same class of anti-diabetic that is being used in the study. So if it was a study using a DP4 inhibitor like SAVOR, the placebo arm would be allowed to have, quote, rescue during the course of the study. And so these cardiovascular outcomes trials, when designed to be most robust for cardiovascular outcomes, tend to then not be so robust with respect to A1C, glycemic efficacy, nor should they be, because we are providing standard of care anti-diabetic therapy. And I think I'll stop there. I talked really fast. I realize we're over.
Well, thank you, Paul. And on behalf of the DIA, on behalf of CITEL, and certainly we want to thank Paul Strump from Lexicon. And I will wish everyone a good day. Thank you once again for participating. And this is Mike Weitz and Zoran Antonevic from CITEL signing off and wishing everyone a good day.
Thanks again, Mike, and thank you, Zoran, and thank you, Dr. Strump. DIA hopes that you enjoyed this online educational experience. We encourage you to visit the DIA website and the CITEL website to view a complete listing of our future educational offerings, as well as to get those slides from CITEL. Please take the time to evaluate this webinar by completing our online survey, which you will be taken to momentarily. Again, DIA is committed to developing quality educational programs that meet the needs of its members, and your feedback is greatly appreciated. The results of our surveys are used to plan future programs. Thank you again to CITEL and to our speakers, and thank you all for attending today's webinar. Enjoy the rest of your day.